Good afternoon. Welcome, everyone. It's a great pleasure to introduce Keith Jerome this afternoon for his 5-year member lecture. And Keith is actually a native of the Detroit area. He told me that's why he's so tough. He made it out of this area to here. He got his undergraduate degree from Georgetown College and then an MD and PhD degree from Duke University, completed residencies in pathology and laboratory medicine here at the University of Washington, followed by a fellowship in the virology division. And he is now here full professor and full member at Fred Hutch. And he oversees a big empire, including the clinical virology laboratory, which provides state-of-the-art testing and innovation and test development for our entire University of Washington Medical Center patients. But he also has a very active basic laboratory program aimed at the permanent cure of viral infections, and I think that's what we will hear about today. And Keith, he is, of course, all that stuff. He is well published, 170 superb papers, reviews, book editors, mentored numerous people who are super successful now, and as we would expect it. And now we are looking forward to your lecture.
All right, thanks, Michael. Is the sound okay for everybody? Yeah, good. All right. So I'm just going to start right off the bat. I'm getting used to doing some of these disclosures. I've done a little bit of consulting for a company called Editas. They're interested in gene editing for various human diseases, including infectious diseases. And then I'll mention several times some reagents that we got from a company called Selectis from France, specifically some of the gene editing enzymes that we've used. So I'll start off just by making the point that it's really important for us at a cancer center to be working on persistent viral infections, and cures for these infections is the theme of my laboratory. And it's important because these, taken together, are a major cause of cancer, and it's been estimated that as much as 25 to 30%, 22% here of cancer is actually caused by infectious agents. So we think about things like human papillomavirus, hepatitis B, HIV, and even herpes simplex. So the first three, very easy to think about them as cancer-causing viruses. 500,000 cases of cervical cancer from HPV every year. Hepatitis B infects 250 million people chronically around the world. The major cause of hepatocellular carcinoma, kills people from that and end-stage cirrhosis. HIV clearly is a huge risk factor for Kaposi sarcoma, 500-fold increased risk, non-Hodgkin's lymphomas, other diseases. Herpes isn't directly an oncogenic virus, but it actually is a major contributor to the last one in that the relative risk of HIV infections is increased over 2-fold by having pre-existing HIV conditions. And if you extrapolate that across the world and where infections with both these viruses have, a lot of the cases of cancer that we think of as HIV have actually been contributed to as the multi-hit process from herpes simplex infections. So the big problem that we have in how we've dealt with these persistent viral infections is that we don't have cures. So we have interventions for HPV. There's a really good vaccine, although it's not used as widely as it should be. For people who are already infected, most people clear the virus or take care of it. But for those who don't, the interventions tend to be pretty invasive. Hepatitis B, again, we have a vaccine. It's not used as widely as it should be. We have good antivirals. We can suppress the virus. But once the antivirals stop, the virus comes back. And that's exactly the same thing we see for HIV. Here, there's no vaccine, and it's still not really clear when there will be a vaccine. We can suppress the virus, but again, that's only temporary, and if you stop, the virus comes back. Herpes simplex. There's no vaccine. A lot of effort's been spent over a long time for a vaccine. It's unclear when, if ever, that will be successful. But we do have pretty good antivirals. We can shorten the course of disease. We can intervene in herpes encephalitis and other things. But again, it's a suppressive therapy at best. And when a person comes off therapy, they're at risk, once again, of recurrence of the virus. So, if we think globally about where we are in therapy for persistent viral infections, we're sort of plucking the top of weeds in our gardens, right? That we can make all the yellow go away, but if we stop this intense effort, everything sort of comes back later. But we should really focus on how good that's been with combination antiretroviral therapy. People living with HIV can have largely normal lifespans now. People with hepatitis B can often reverse much of their liver damage and sort of reset their liver and so forth back to where it was before, even with a course of relatively limited duration. Suppressive acyclovir can prevent recurrences, reduce shedding, and thus the risk of transmission to others. But for all these, as I said, once you stop, the virus comes back. So we'd really like to think about cures for these viral infections. And the big hurdle, the big stumbling block that we have is that all of these that I've talked about have long-lived DNA forms somewhere in the body that persists. Okay, so for HIV, the virus, it's a provirus, or it's a retrovirus, right? So it gets integrated into long-lived memory T cells. For hepatitis B, there's a form called covalently closed circular DNA that lives in hepatocytes and kind of slowly replenishes itself and has a very long half-life. For HSV, the virus establishes latency in neuronal bodies. So we'll go into the life cycle a little bit more, but it infects the mucosal surface and basically establishes latency in the nerve bodies that innervate that region. And so to cure any of these, you need either to, one, get rid of the infected cells. Maybe you could do that for HIV. You really can't get rid of your neurons, and you can't get rid of all your hepatocytes. So you either need to get rid of those cells or get rid of the virus within the cells. And the argument that I'm going to make to you is that if we could do that, get rid of it or get rid of most of it such that we reduce reactivations of virus, this would be of tremendous clinical benefit to patients. Okay, so let's look at the biology of herpes simplex just a little bit. So first of all, it's a very common infection. About half the people, it depends on what socioeconomic group you look at, but you might say HSV1 seroprevalence is about 50%. In the United States, HSV2 is about 1 in 6 people are seropositive. And the virus infects in the mucosal surface. Here we go. Mucosal surface, it starts some infection there, and it gets access to nerve endings, travels down the axon to the nerve body, and this is where that process of latency is established. So the virus goes dormant there. It could live for weeks, months, even years, until eventually from some either stochastic event or some sort of insult that's happened to the neuron, the virus reactivates, comes back out, and is able to reestablish infection here. So we can get asymptomatic shedding, we might see a lesion, but the virus now has this opportunity to transmit to others. So this latency happens in the trigeminal ganglia, in the head, along the spinal cord. For HSV2, would happen in the dorsal root ganglia. It can also happen in autonomic ganglia like the superior cervical ganglia, and we'll talk about that a little bit later in the presentation. So these reactivation events cause ulceration, shedding, can cause serious things like keratitis that can lead to blindness. It can cause neonatal disease that can be fatal to newborns. So there's actually some severe disease that can be caused here. Typical recurrence rates anywhere from none to monthly, depending on a person's biology and their immune response. And for a lot of people, how this infection is perceived by people really varies. For some people, it's a nuisance, and they don't think much about it. For others, it's a very big deal. And one thing that I always throw out is that the last year that acyclovir was on patent was 1994, and people spent well over $1 billion on that, even though the drug is not all that good at preventing recurrences or treating recurrences. So it's something that people do care about, and there's a need. Let's look a little bit more at the latency. So I told you that the virus is going to lie dormant in these neuronal bodies that innervate the peripheral sites of infection. So latency can be established either in a sensory or autonomic ganglia. I mentioned some of the names of those, like the trigeminal, superior cervical, and dorsal root. The first two, remember that the trigeminal is a sensory ganglia and the superior cervical is an autonomic ganglia. Those are going to come up as I get into the data that I'll show you. But just for perspective, a typical ganglion might have anywhere between 10,000 and 20,000 neuronal bodies in it, okay? Of those in an infected ganglion, maybe 10 or 20% actually have herpes. I'll actually show you data for this in our hands. And each one of those that's infected might have somewhere between 10 and 50 copies. So those are all at risk sort of constantly for reactivation, all those episomes. Any given one might reactivate at any given time. And data generated from this group, Josh Schiffer and his colleagues, suggests that reactivation is sort of always happening at some degree or another. And the question is sort of how much happens to be coming out at a certain time, and is it actually being controlled by the immune system at the periphery? So this reactivation is very common, but the outcome that we perceive, is there a lesion, is there shedding, is very, very different depending on the biology. But we do know that this initial seeding is very largely driven by the ganglionic load. Having more virus is bad. You just have more chances for more shedding, more chances for lesions, more disease. So all these things put together actually make this a really nice target for therapies that we're directing toward the persistent DNA form. So I mentioned HIV, I mentioned hepatitis B, I mentioned herpes. HIV scattered all over the body, and one T cell in a million, right, can actually reactivate. How do you find that? Where is it? Is it in the lymph node? Is it in the gut? Who knows? Anywhere in the body. Hepatitis B, we know it's in the liver, but there's a fantastic number of cells in the liver, billions and trillions of cells, and they all have hepatitis B. It's just a massive problem to try to deal with. Herpes has a relatively few cells that cause all disease, and they're all in one place in the body. So if we had an effective intervention and a way to get it there, this is the kind of target we would want. Before I go on, I showed you that thing about people spend money on acyclovir in 1994. We used to get questions about the work, say, why are you working on trying to cure herpes? There's serious diseases to deal with. Nobody cares. I actually got essentially NIH grant reviews that said that. So thanks to Linda Oseso in working together at Andewald, we used some of the methodology that had been developed for HIV cure to ask questions about, for people living with HSV, do you care about cure? What would be good about a cure? What does a cure look like? Would you be in a study? Those sorts of things. And what we found was really quite dramatic. And first of all, anything you can name about cure is generally extremely desirable for people who participate in this. It's a little bit of a self-selected population, but you can see that almost everything really comes out very, very positive. And most importantly, almost unanimous, is people don't want to transmit the infection. It makes a lot of sense. We see this for other viral infections as well. And even more so, if we do get to the point where we can actually start thinking about doing some of these interventions in people, people are excited to be involved in trials. So even in early phase trials, even if cure is a long way off, there's clearly a lot of people who are willing to participate. So I think this sort of data, and more recently we've compared perceptions of HSV cure to HIV cure, and I think one thing you really just can't argue anymore is that nobody cares about this. So I'm really grateful that we were able to get this work done. All right, so how are we going to do all this? What we're going to use is targeted gene editing, CRISPR-Cas9. You've heard about all this stuff. So I think most people are familiar with it. I'll give a really quick primer. It's not that complicated. The idea is you have some sort of nuclease. Everybody knows restriction nucleases, target, a specific sequence of DNA, maybe 6 or 8 base pairs long. These are rare cutting endonucleases. They just target a longer sequence, right? So you're looking for maybe a 15, 16, 20 base pair sequence somewhere in the genome, in the DNA that is exposed to this. And if the enzyme sees it, it sits down and it causes a cleavage, induces a double-strand break. Some gene therapy folks try to repair this by homologous recombination, in which case you try to put something in there. So you might try to repair an inborn error of metabolism, for example, with this. More commonly, and for our purposes, we are allowing this to just be repaired by non-homologous end joining. This is actually the major repair pathway that happens most of the time in mammalian cells. And some fraction of the time, this is imprecise, and so it causes a disruption of the gene. It knocks out the gene. And in fact, there's a lot of tweaks to make this happen. So you get pretty efficient gene knockout. by using this approach. And so our initial idea was that if we targeted these enzymes toward the virus, we could knock out an essential viral gene, an essential viral protein, and thus the virus, even though it was still there, wouldn't be able to replicate, wouldn't be able to cause disease. It would no longer be a problem. Functionally, you would have a cure. So there's lots of enzymes that can do this. Everybody, I think, has heard about CRISPR-Cas9. This is, all the rage. There's lots of really cool things about this. Really easy to retarget. So you just feed it a new RNA, and that'll tell it, which sequence to go look for, essentially. So you don't have to design anything about the protein. But it's important to remember, and I try to stress this in all my talks. There's actually a lot. There's another, several groups of enzymes that can do the same thing. There are zinc finger nucleases a lot of people have heard of. There's tau effector nucleases. And there's something called a meganuclease or a homing endonuclease that can do the same thing. Look for a specific sequence, induce this double-strand break that'll lead to mutation. So everybody works with this because when you have a target, you only have to change the RNA to sequence that target. If you want to target something specific with any of these, you need to change the entire protein. You actually have to change the structure. And for some of them, they're modular, for the zinc fingers and the taus, so it's not so bad. For the meganuclease, you're actually modifying the structure of one domain that mediates recognition and cleavage all at the same time. So it's actually a complicated task. And we have some great collaborators who can do this, but it's not easy. And so these don't get used as much because you have to put months of work up front just to have an enzyme that can cleave something. But I'm going to show you that effort spent up front is actually really useful. And for applications such as ours, it's actually critical. Okay, so where does gene editing for viruses stand? There have been well over 100 papers now that have evaluated this, mostly in vitro. So I get a culture system, I grow a virus in there, I throw my nuclease on there, and I go, hey, I mutated the virus. That was very exciting 6 or 8 years ago, and it continues to be demonstrated over and over to this day. And you can do this very well. I mean, the efficiency of this has gone up. Some people are able to actually eliminate virus out of a culture now, HIV or something like this. So it actually does work. But the challenge has been taking what we can do in a dish and turning it into an in vivo therapy. So you've got to deliver these enzymes to the right place in the body. You've got then to start to make the efficacy go from 10% to 20% to 30% till you get into a range where it's actually useful. Then you have to show that it's safe, right? Does the animal tolerate this? I've just put a gene-cutting enzyme into an animal. Is that okay? So in contrast to all those papers for doing this in vitro, there have been 3 that have done this in vivo, 2 for HIV, and then 1 from our group that I'll touch on very briefly, where Martine, who's here, has done this for herpes simplex. In order to do this, we were fortunate enough to establish a collaboration with a company called Selectus, so I mentioned early on, and they have developed a series of enzymes that target sequences within HSV1. I'll talk mostly about 2 of them. One is called HSV1M5. I'll sometimes just call it M5. One is called HSV1M8. I'll call it M8. M5 targets the UL19 gene of HSV that encodes the major capsid protein. M8 targets UL30 that encodes the catalytic subunit of DNA polymerase. We also have another one that I think there's actually one figure up there called HSV-M4. It targets ICP0. It's unique. We were interested in that because ICP0 appears twice in the HSV genome. So I'll point that out when we get to it. We also have a control called NV1. We just called it that for non-viral control, non-viral one. It actually targets the cellular RAG1 gene locus. That's not relevant for our studies. So these induce a double-strand break. For a few experiments, especially early on, we'll co-express these with a second enzyme called TREX2. Meganucleases leave a 3' overhang of 4 base pairs, and TREX2 trims those off, and so it lowers the frequency of precise repair and makes there be more mutation. In later experiments, we've shown you don't really need it, so it's mostly come out. There's a couple experiments that include that. A couple of years ago, we did what I was just kind of dissing. That is, we showed this in a culture system, and Martine set this up, and it actually was not a simple system, even though doing things in culture you might think is simple. Establishing herpes latency in a culture is difficult. So this was done in primary fibroblasts that were infected with HSV. You have to do this in the presence of interferon and acyclovir, and then you wait a certain amount of time. And then the virus enters a state that looks pretty much like latency. If you look at the genetic program, herpes during latency expresses one transcript only, LAT. We'll come back to that. It's really important. And you can show that this model recapitulates that. And then if you reactivate the virus, you see immediate earlies and then earlies and so forth. So it really looks like latency. So we asked, if we take those latently infected cells and treat them with these meganucleases, can we induce mutation? And the answer was yes. We put them in at the time using a AAV2 vector. We'll talk about AAV a little bit more. But we can induce mutations. This was done by clonal sequencing. So we actually took individual clones out and sequenced them, and we showed a variety of mutations. So yes, a meganuclease can target a virus, and that was relatively new at this time. And as I said, this has been supported many, many times since. But the big challenge was taking this and moving it into an animal. And so the system we set up that Martine really perfected was to take old models of herpes virus latency that were done in mice. And for this, you infect mice with the virus here in the face. And then you wait a month or so, and the virus travels down those nerve endings, goes to those cells, those neuronal bodies, and establish latency. So the virus goes dormant, and again, the genetic program is exactly what it is in human beings. The only difference in a mouse is that it won't reactivate and come back out. But everything else is the same. And then we can take those latently infected mice and ask, if we put in our adeno-associated virus vectors that carry the coding sequence for these meganucleases, can we get any gene editing of HSV? So these are what the initial early vectors looked like. We had, if you have an adeno-associated virus vector, you take out all the genes of adeno-associated virus, except for you just leave the inverted terminal repeats, and then the whole rest of the middle is available for your transgene. So here we used one of our meganucleases, NV1, M5 or M8. We put that together with TREX and a little fluorescent marker so we could see everything that was happened. And we didn't know if AAV would work, but we had heard that AAV was thought to jump on molecular motors and run down axons right to the same nerve bodies that herpes lives in. So we put this into the whisker pad of mice with a little tiny injection, just a little bit of fluid. And the answer was, yep, it goes right to there, and you can see plenty of AAV in these ganglia. So it seems like we had the delivery part worked out. And in fact, if you did this and then later took those ganglia out and asked, have we observed any mutation of herpes? The answer was, yeah, actually we do. This is what we'll call a T7 assay. And essentially you just amplify the area where your mutation is supposed to be, and then you treat it with this T7 enzyme. And if you see bands down below there, these are cleavage bands, and they suggest or they demonstrate that you've had mutation there. Okay. So the more intense the band is, the more mutation you've had. You can kind of quantitate it, get a general sense of a little bit, a lot. We'll put a number on it, but you have to take that with a little bit of grain of salt. And then you can confirm that sort of mutation by taking those same regions and suggesting it to next generation sequencing. And so what the take-home message of this was, yeah, you can actually get gene editing of latent herpes virus genomes in vivo at a pretty low frequency. About 2 to 4% of all the latent herpes in there shows evidence of mutation. Okay, so this is really cool. It's the first time anybody had shown this in vivo, any kind of gene editing for any virus. You could do it in vivo, but you're not really at the level that's probably going to help anybody. Okay, so but there was some good news out of that. First of all, you could put a lot of gene editing enzymes into animals with an adeno-associated virus vector, and the animals looked just fine. They didn't seem to mind it the least. There's no evidence of any inflammation. There's no evidence of neuronal death. These are the ganglia that look completely normal. And if you look genetically at the sites within the mouse genome that are most similar to the herpes site you're targeting, we saw no evidence of any off-target mutation, okay? In fact, the only thing that shows up in this whole thing was just one site that happened to have This is probably just a technical aspect of the sequencing, but appears both in the control and in the experimental group, a little bit of mutation. So it really looks like this is safe, it's tolerated, no evidence of genotoxicity or overt toxicity. It just needs to work better. So the first thing we thought of, we're doing all of this with meganucleases because the project started before CRISPR-Cas was actually figured out. Maybe CRISPR-Cas9 is the answer. So let's try that. We can put those into our AAV vectors and see what happens. So we went back to a culture system. We'd gotten a little bit smarter now and actually had learned how to culture neurons so we could have latently infected neurons, the real cell that we need to do all this in, and treat them with CRISPR-Cas9, and we identified a bunch of good guides. So if you know how that works, you have to pick the RNA for exactly what part you want to target. Not all created equal. So you find some good ones. And now, hey, this looks like it's pretty good. I mean, look at this one. It's got nice dark bands there, and it comes out, your guesstimate is it's almost a 25% mutation. It's all pretty nice. They don't seem to work quite as well for these, but we've got a good one. So let's go with that and see if we can recreate at least what we saw before. If not, maybe we've got better mutation in vivo. And the answer is, amazingly enough, it actually doesn't work. So we do the exact same experiment. We establish latency. Now we put in, we've now improved our AAV vector. We've got actually a better AAV vector called AAV1. We'll talk about AAV serotypes. You put all this in, and then let's see, we can quantitate HSV in there, show the animals are infected. We can quantitate that the AAV goes where it's supposed to, the one that's carrying the different guides and the CRISPR-Cas9. But we see absolutely nothing in vivo by T7 assay. So if we go and do the next generation sequencing and look really hard, yeah, there's a little bit of mutation with that one good guide, that 26 that I showed you, but it's almost nothing. It's 0.2%, 0.3 of 1%. So we've actually taken a tenfold step backwards by going to CRISPR-Cas9. So we spent a lot of time trying to figure out why that was and what's the problem here. We tried things like changing the promoter that's driving Cas9. Even though that was a promoter that seemed to work well for the meganuclease, maybe there's something better. Didn't help. Tried different guides. We had those other ones, tried those. Didn't matter. Tried a bunch of AAV serotypes. I won't really go through all the data here because it's mostly negative. But the answer is no matter what we do, we can never get beyond a few tenths of 1% in any of these experiments, right? There's a 0.3%. There's one that's about 0.5. But clearly nothing like where we need to be. So one thing we started to consider in making this better is our selection of vectors. So I'll get a little bit into adeno-associated virus vectors. I said you can take this whole AAV and you can lop out all the actual genetic material that makes up the virus and use that whole size for your transgenes. But all that needs to fit within a very defined capsid of AAV. So it can only fit so much, and that is the absolute limit. And if you go past that even a few %, the virus just won't build itself anymore. So it's a hard limit. Unfortunately, Cas9, the Cas9 people mostly use, the strep pyogenes Cas9 won't even fit, okay? But there are other good ones. We use one called Staph aureus. It's a little bit smaller and just happens to slip in under the wire so you can make it all fit. And it looks something like this. So here's the size of what you could fit. So there's our ITRs. You're allowed to have about 4.5 kb. If we put the SA-Cas9 in there, it's about 4.1 kb. So we can play a little bit with small promoters, all the other stuff you need, polyAs and so forth. So it fits and it's fine. But there's actually a trick with AAV you can do. If you happen to have a small transgene, you can take advantage of the ability of making it a palindrome and make it fold onto itself. Now, why would anybody care about that? When AAV goes into a cell, either a wild-type AAV or a vector, it's actually a single-stranded virus. So you only have one strand. So you need to actually undergo a process called second-strand synthesis before you can make the episome that can actually express the transgene, okay? And the virus doesn't carry any machinery to do that. So it goes in and sort of sits around and essentially hopes for the cell to complete that process for it. So that is slow and unpredictable, okay? But if you've come in with a vector that has this palindrome that I mentioned, as soon as it goes into a cell, it essentially folds back on itself and that second strand synthesis happens immediately. And so what's known to happen is you get very rapid expression of your transgene, and you get very high level expression of your transgene. So those seem like very good things for us, but Cas9 is way too big, right? But I don't know if you remember back to that picture I showed, and they're all almost pretty much to scale, that Cas9 is that big protein, very complicated. The meganuclease is a little tiny protein, and actually has a little tiny coding sequence, and actually fits. So these things are called self-complementary AAV. This is single-stranded AAV. And these have this coding capacity of about 2.4 kb, and it turns out the meganucleases fit. So we wondered, what if we take what we have, go back to the meganucleases, but try to ramp it up by using this self-complementary AAV? Well, first of all, we just put a fluorescent transgene or a fluorescent transgene into cells and said, is self-complementary really better than single-stranded in neurons? This is 28 days after transduction, and I think you can probably already see that at pretty low levels of viral input, with the single-stranded AAV, there's some dark cells, but there's a whole lot more here with the self-complementary. These are all the neurons, right? So we'd like to cover all those. We're only getting a subset, but it's at least in the right direction. And if you look 56 days after doing this, I think it's a pretty dramatic difference now that the self-complementary is clearly better than the single-stranded. So we felt like maybe we were on the right track, and this might be something that was worth trying. All right, so what happens then if we take self-complementary AAV and we introduce a meganuclease? So same experimental setup, we latently infect mice. Here we wait for 33 days, and then we treat them with AAV. That introduces our meganuclease, and we'll wait some time and then analyze to see if we saw any gene editing. First of all, I can tell you that we need to make sure there's herpes in these mice. Yep, the herpes is there just like it's supposed to be. Did AAV get where it's supposed to go? Yep, those are almost exactly equivalent. There's about a 50% difference there. But do we see mutation? And the answer is yes, regardless of whether we use AAV1 or AAV8, so two different kinds now that I'll come back to. We actually start to see pretty robust mutation in vivo now, okay? These are very clear bands in vivo, and now we're looking at 6, 7, even 8%, okay? So 8% of the virus remaining in these mice has virus in that, or has these mutations in it, right? So we're starting to feel like we're at least moving in the right direction. About the time we were doing these experiments, we had another insight. And that goes back to something that I talked about at the beginning, that herpes infects not only these sensory ganglia, the TG. That's what everybody in the herpes field is always thinking about, the TG for HSV1. That's where the virus goes. But it also goes to these autonomic ganglia. And we realized that we have access to those in our model system. And so we said, well, let's just see what's going on in those. And one reason people haven't looked at them so much is that if you just look at the viral load in a superior cervical ganglia compared to TG, it's supposed to be less than the SCG. So some people said, well, that means it's not important for human disease. Other people, and unfortunately Larry's not here because he's one of these folks, has argued that the autonomic ganglia are actually one of the major drivers of recurrent disease and very, very important in that. So we looked in our model system, and in fact, we recapitulated what people had reported before, is that there's a lot more herpes. Here it's expressed, this is a digital PCR assay just for herpes. So there's a lot more virus per 100 neurons in the TG than there is in SCG, but there's clearly virus there. But what was interesting is that if you look at and quantitate your AAV, it's the converse. We were really surprised to see this, is that AAV actually loves to go to the superior cervical ganglia. It's actually like, several-fold higher. And so we started to wonder if there's less virus but more AAV, more transgene to target it, maybe we'll see more gene editing there. Maybe this can test this idea that it's very simple and simply more is better. So you can see the AAV to HSV ratio is very advantageous in SCG compared to TG. And then if you look in those SCG and say what levels of mutation am I getting, in this experiment where we saw about 2% in the TG, now we're seeing 8 to 10 to almost 12% in the SCG. Okay, so now we have a new tissue, what's thought to be a clinically important tissue, and we're actually doing better, and we can really start to use this to optimize our therapy. The other advance that we had actually came out of my good fortune to also work in the HIV field, and as you can tell by now, I've sort of, rather than give an overview talk, I've really today wanted to really drive down or take a deep dive into our HSV work. But one thing we did learn from HIV, the people doing gene editing all started out kind of like we did, saying we're going to find a locus and we're going to induce a mutation there, and it's going to be repaired by NHEJ, and you'll have a deletion or an insertion there, and the gene will be knocked out. But some folks were initially targeting the long terminal repeat of HIV. So remember, there's one of those on each side of the integrated virus. And they found if they targeted that, not only did they see mutations there, but even more frequently what they saw was the entire virus was just excised, right? So when the cell tried to repair that chromosome, it just found those two ends, stuck them together, and HIV was gone. So we started to wonder if that might happen here. That is, if we targeted HSV in two places, maybe we would just get rid of that intervening sequence. That'd be a really great way to knock out the virus. Or maybe because now you've got a floating episome that's just floating around in two pieces of different sizes, maybe it would just be degraded and go away. In fact, that turns out to be the case. So in this series of experiments, we established latency again, and then we came in with AAV8 vector that we'd shown in the previous experiment actually works pretty well. But now half of the AAV8 expresses M5, and the other half expresses M8. And now we can look and say what happens in those experiments. And so you can look at the two target sites. So that's what we're doing here, and it's pretty hard. From this angle, I can't see. There are some bands. I don't remember where they are now. I think they're in the M5s, yeah. It's a little more quantitative if we look down here by the NGS. Yep, at each of those target sites, we see mutations at those target sites. It ranges up in this sort of 8 to 12 range at best for in the SCG. It's about 2 to 4%, just like we've seen before for M5. A little lower for M8, but clearly we're having mutation. But it turns out that the trick here, or the critical factor, is not looking at mutation of the virus that's left over, but it's actually in saying how much virus is there at all. Because what actually is happening is a substantial fraction of the virus is actually being lost when we do this. And we see this in several places. So we have about I think in this series of experiments, about a 60% loss of virus in the SCG and about 40 or 50% in the TG. So we're actually getting destruction of the virus. And when we look at the mutation, we're sort of seeing the remnant virus, and some of it happens to have, notice that, yeah, it's been cleaved, it just happens to have successfully repaired, but most of it's actually getting lost. So we've really been driving forward along this pathway in recent days, including really trying to optimize treatment with a single meganuclease. So, what was the answer there? Was this successful because we used two enzymes or because we're really getting the AAV types figured out? And we looked at that very carefully. And in fact, if you optimize things and look at a wide variety of AAV serotypes, including some that are synthetic and never exist in nature, things like RH10, for example, I'll just cut to the chase that ones like RH10, which are very, very good at delivering transgenes to the SCG, even as a single enzyme, can show a substantial loss, about 70 or 80% elimination of herpes from the SCG. And then what looks like a pretty convincing trend toward loss in the TG as well, but it didn't reach significance because we didn't have enough animals in the control group. All right, so kind of where we stand now is if you take all this together and you take a really good AAVs and you combine it with two enzyme therapy, to cut straight to the chase, we're getting a greater than a log decrease now in viral loads in the SCG and greater than 50% loss in the TG. So we're really getting rid of that. Now maybe I'll just, for purposes of time, send you, same experimental design. But now if we look in the SCG and the TG, here is the reduction in virus, again, about log, and then about 50% here in the TG. And if we look at mutation, it's not all that good, right? So this is why we're a little bit on the wrong track. I'm glad we finally looked at how much virus is there overall. So the other aspect of this is, of course, we want to get rid of the latent virus because we don't want virus to reactivate, right? I mean, that's what we really want. So we decided to do an experiment in which we took these cells that we've treated, an aliquot of them we've analyzed to show that there are fewer latent genomes. What happens if we try to reactivate the virus out of those? And if you put them in culture and stress them a little bit, virus starts to reactivate and actually come out of them. And we can show that you get, it's not an enormous amplification. We get a several-fold amplification of virus if you do that. So how much comes out in a treated versus an activated cell? We would expect that a treated cell should have at least a log decrease, just like of reactivated virus, just like it does in its viral genome. And in fact, that turns out to be exactly the case. Now, these are all the reactivated viruses. And now if we look here in HSV genomes per million cells, you see this 50% loss again here in TG and over a log in the SCG. So we're actually moving in the right direction that, although these mice won't spontaneously reactivate and send things back to the periphery, in culture they do reactivate. We're able to impact upon that process. All right, so in the last few minutes, why are we only at 90%? So, we're very happy. We were at 4%, now we're at 90, so we're definitely moving in the right direction. But, we want to be at 99 or 99.9% or get rid of all detectable virus. Well, we know that the AAV serotype that we use and the way we put it into the body actually makes a big difference. I didn't talk too much about that, but we've done a number of experiments evaluating different AAV serotypes. 1, 8, PHPS, and RH10 kind of made a short list of serotypes that have looked really good in preliminary experiments either that we've done or others. Turns out they like to go in different ways. AAV1 works the best via the whisker pad. Most of these others, it turns out we can just put into the blood, so either in the tail vein or into the retroorbital vein, and they actually work better that way. But it's very clear that different ones have different, kind of maximal effects. So here's this RH10 that I showed you we used. Kind of we think of it as our optimal one. So here's SCG mutation by NGS. So that 30% mutation is going to be on top of all the loss that I showed you before. So if it's been reduced by 90%, we're getting another third is mutated. So 93% is gone. So it's all good. But what's the difference between these? And is the answer just we need to find the ones that kind of get there the most? Or is it a more complicated situation? So we know that AAV serotypes have different tissue tropisms. This is just a schematic of AAV 1 through 9. These are sometimes thought of as the naturally occurring AAV types, either in humans or other primates. And then there's all these other types that have been built by either reshuffling them, things like RH10, or ones that we've built where you actually derivatize the surface and make them bind to what you want. And this got cut off. But they actually, the point of this was to show that different ones actually they have different abilities to actually go to the ganglia and then actually to express their transgenes. So even once they get into the neuron, they have different abilities to release their viral DNA and actually make their transgene. So we need to really study what's going on. And one potential issue is, we already know that there's a difference in efficacy between sensory neurons, the TG, and our autonomic neurons like the SCG. But it's even more complicated than that because if you think about a sensory neuron, it has mechanoreceptors in it, it has proprioceptors in it, it has nociceptors in it, and you have subtypes of autonomic neurons as well. What if herpes likes to go to certain types of neurons, and certain AAV serotypes like to go to certain neurons? Then it's really important to know, how much do those preferences overlap so that we can get our transgene to the right cells. And so we were fortunate that Dan Strong in the lab had a lot of success using the 10x genomics platform to perform single-cell analysis from our animals and really ask, where's herpes and where's AAV going? So Dan gave a couple of great talks the last couple of weeks on this, so hopefully you heard some of that for the technical details. But in a nutshell, it's all based on these little coded beads. And they're all barcoded with a little genetic barcode such that each bead has a unique identifier. And then there's also this unique molecular identifier that many of them are on this bead that we'll talk about in a second. But essentially you flow these beads in through this microfluidic device. They get combined with your cells and your RT-PCR enzymes. And then they get encapsulated into a lipid droplet so that each droplet contains one cell and one bead. Then the cell gets lysed, reverse transcription happens, and a cDNA library is made. But now everything in your cDNA library that was from that cell has that barcode, right? Okay, so you know exactly which cell your read came from. And there's also this molecular identifier allows you to rule out PCR amplification, so you know it was a unique starting molecule. And then you can kind of throw all this together and sequence it on Illumina. And by reading out that barcode at the beginning of every read, you know that, for example, cell 1 had a little bit of gene 1, but a lot of gene 2 and very little of gene 2000, whereas gene 10 or cell 10,000 kind of had the converse pattern. And all this happens on this chip, and it can be done here at the Hutch genomics resource and very powerful technology. So we did an experiment in which we infected mice for 60 days with HSV1, established a nice robust latency. And then we treated them with our 4 leading candidates of AAV. In this case, no gene editing, but each one has a genetic marker. And to make it easy for ourselves, we put in the fluorophores we like to work with. So M. scarlet, EGFP, DS-Red, and BFP, but each one correlates with a specific AAV serotype. We let those express themselves for 3 weeks. Then we collected the tissue, we pooled all the TGs, we pooled all the SCGs, we dissociated, purified the neurons, made the libraries, and analyzed it, okay? So out of all of this, we were, in theory, we were hoping to get about 20,000 cells to analyze if there were no losses at all. There were some losses. But what came out of all this was 2,319 neurons from TG and a little over 2,000 from SCG. For each of these, we were able to identify expression levels of over 5,500 genes for each cell, and at a very good depth, so about 100,000 reads per cell, which means we can actually have a good sense of dynamics, what's expressed a lot, what's expressed a little. So the first thing you can do with this data is define neuronal subset clusters. And so you get thousands of genes, and you cluster all this using principal components analysis, and then it gets presented on this t-SNE plot, so you can actually visualize these complex, multidimensional groupings in 2D that we can understand. Well, the first thing that falls out is that SCG and TG are completely different tissues. And we knew that, but it made us feel good that that falls out immediately in this analysis. But it also falls out that there are subsets of neurons. We sort of knew this already. It wasn't new. But there are subsets of neurons, so we can break them up. And in our analysis, we defined 10 unique populations of trigeminal ganglion neurons and 5 populations in the SCG, and they're color-coded over here. All right. So this is what this data actually looks like in a more complex form. I know you can't possibly see it, but along the top are the groupings from SCG and TG. Each one of these along the axis here is a different gene, and then these are individual samples. And so you can, individual neurons, excuse me. So you can see that, this is the pattern for group 2, sort of doesn't have these up here, but it's got a lot of these and none, and it's very distinct from pattern 1. This is pattern's unique, SCG and the TG are quite different. So you can actually tell these things apart. Just to give a sense about how distinctive these populations are from each other, if you do a heat map just to say, how well does population 1 agree with, our own population 1, it's a perfect correlation, right? So that comes out in bright yellow. But how close is our population 1 to population 2? Well, yeah, it's a SCG neuron, so it's got some similarity, so it's a little bit yellow. So if you think about how this works, what this is all SCG neurons do have some similarity to each other, even though they fall into these populations. They're quite distinct from the TG neurons, so we see this nice line right across here. So it turns out that 3 groups had done this kind of analysis, either using 10x or other similar technologies in TG. And I just want to show you that we were very happy to see that at a global sense, our groupings of neurons actually look a lot like what was reported in literature. I have to double check this. I think this one was actually 10x. It actually seems to correlate pretty well. Here's the SCG. They don't correlate with anything else, but like our TG1, tends to correlate with their C1 and so forth. They defined 13 compartments. We had 10, but you can see there's actually nice correlation. So we're actually on the right path here. So importantly, it turns out that our hypothesis was supported that herpes and our AAV serotypes are non-randomly distributed across neuronal subtypes. So, we were really fortunate we were able to find HSV lat in these, okay? A latent HSV makes 1 transcript. We were able to find it, okay? And it turns out, not surprisingly, HSV is more in the trigeminal ganglia and the SCG. We showed that by digital PCR several slides ago, right? We also found some interesting things. For example, our AAV1 is actually pretty good at going into neurons in the TG. Doesn't go to the SCG well at all. 8, no wonder we sometimes liked 8 and sometimes liked 1. They were sort of going to different ganglia, right? And RH10, which we really liked quite well, it goes to both reasonably well. So that's why that one's done pretty well in some of our analyses. So the big money question is, how do all these things tie together? What happens if you superimpose these over those? This just shows the distribution, the preference of herpes and AAV to the different subtypes. But this is the figure that I wanted to show you, where we superimpose now where AAV is, where herpes is. So in this diagram, so here's our subpopulations again. Herpes positive neurons are here in green. AAV positive neurons are in red. And then neurons that contain both HSV and AAV are the diamonds. So you can see lots of those here. Now, it took us a second to look at this. I was a little discouraged when I first saw this. I think Dan actually had this figured out. Remember that we've combined four experimental groups into this, which means that only a quarter of the animals were ever exposed to any of the AAV serotypes. So you're only a fourth of the neurons ever had a chance to get PHPS, for example, or RH10, okay? So you've got to sort of think about what would this look like if I had 4 times as many of the AAVs, okay, if the entire read run were used for that. So if you do that math and look at this, it actually is very satisfying because it confirms what we observed. And that is PHPS in the SCG, we're hitting about 80% of all the neurons that have herpes. We're getting a transgene to them that we can measure, okay? Similar amount with, with RH10 or here with AAV8. Those are the ones that really work well, and we've seen great gene editing in the SCG. In the TG, it hasn't been quite as good. Some of our best data has been with AAV1. Our best data, but we've had much worse gene editing there. And the reason is we're not as effectively getting to the infected neurons there in the TG. So we have some work to do there. So just to wrap up, I hope that you've been convinced that we can perform targeted mutagenesis of latent HSV in vivo. And where it stands now, we can eliminate over 90% of HSV from the superior cervical ganglia and over half of it from the TG. You can do gene editing with a lot of different kinds of enzymes, but I would encourage you to be open to other classes that aren't CRISPR-Cas9, because the fact that they're small and the fact that they can be targeted or packaged up into specific gene delivery vectors may be very, very beneficial to these sorts of applications. If you haven't tried our single-cell RNA-seq and you have an appropriate application, it's a very powerful technique, and we're very interested in optimizing future efforts in gene therapy with this technology. And I think the take-home message for our work from the single-cell work is that we're going to need to use combinations of very carefully selected AAV serotypes in order to be sure that we get to all the relevant neurons. But if we can do that, I think we can actually come close to eliminating a virus from infected ganglia. So with that, I have to thank a lot of people, a lot of work here, and very much a team effort. Really, everything was spearheaded by Martine, so she's right up here first. And in all the animal work, she's done together with a great team of Michelle and Nori, who have worked really hard at this. I mentioned several times Dan Strongen, who did the 10X work. It's been fantastic, and I didn't mention, and I should have, how important the data crunching and the genetic analysis of that data by Pavitra has been to all of this. And we could give a whole talk just on data that she's generated and hypothesis. The PCR is led by Mei Li Huang and her technologist Larry Stensland. Alex and his team did all the next generation sequencing, and it's been very critical to getting us on the right path. Dan and Beth are sort of our AAV vector team, done a lot of design, a lot of growing of the virus. Tom has made sure that everything we do is appropriately meets all the regulatory standards and stays on track. So thank you for that. Mary keeps me online. I want to thank the Genomics Corps and Selectus for providing us enzymes. So with that, thank you. I'll take any questions. Oh yeah, hi. Yeah, good.
I feel like other times you might have included conversation about number of doses. Is that still a factor that might be influencing how many you're asking for?
Well, we really started that, again, thanks to Josh and his team, with the idea that, a dose is getting 5% or 10% or whatever. Now we're looking at a situation where a single dose is getting 90+%. So we may not need to do that. I'm actually cautiously optimistic that with better, I think we have a really good reason why we're where we are and a good pathway toward making it better. So we may not need to do that, which is actually good because, if you treat with an AAV vector, you do induce an immune response. There are ways to get around it, but it makes repeated dosing more difficult. And so it's actually nice. If we can do this all with one dose, that will be great. And it may also give us, an improved safety profile as well. Any therapy you want to do for as short a time as possible, just on general principles. So, we may come back to that, but I hope we don't have to. Yeah, Floyd.
If you reactivate herpes from a neuron, does that in any way impact the susceptibility to AAV?
We haven't looked at that specifically. So if a neuron is actively reactivating herpes, might it not want AAV to get into it?
Or more easily?
I would suspect that the more easily, certainly for wild type AAV, AAV is a replication defective virus, so it needs a helper virus. And although in its name, it's adeno associated, so it goes along with adenovirus. In fact, what it usually goes along with is herpes viruses. So it can jump onto those and work better. I don't think evolutionarily, I think it'd be the converse to be true would be very surprising that a cell with herpes in it is somehow refractory, and I think we have lots of data that's not the case. Yeah, Daphne.
Kind of along the same lines, though, what about the chromatin state of the viral genome? So in the different neuron types, there may be different chromatin states of the metastasis inhibiting the enzymes from finding their targets, and reactivation would allow a change to some
Yeah. Yeah, so that's something we've explored in the past, actually, and that's certainly the case. So in our culture models that I just touched on really early, once I started the data, we demonstrated, I think, in what I showed you, maybe 4 or 5% mutation rate with a meganuclease. In fact, what we found is that we treated with certain chromatin modifiers, we could double that. So trichostatin A, I think, was one. So, HDAC inhibitors, these sorts of things. We actually tried a series of them. We get for some of them, we get about a doubling, very consistent. So I think chromatin structure definitely does matter. And in fact, we're actively looking at chromatin structure at our target sites to make sure we're not looking at a highly repressed area. It's one reason to think about LAT, for example, in the ICP0 locus, which is thought to be more open. We haven't demonstrated, in our limited experiments, we haven't demonstrated that those enzymes are clearly superior yet. So, it may or may not be true. I was more concerned about that when we're having very low frequencies. Now that, this all seems to be working very well, it may not turn out to be as big an issue as other. It could be, yeah. And if that's the case, then we'll be right back doing that, and we'll come and talk to you. Oh, one in the back, Andy.
Yeah, I was just wondering, coming back to maybe having to treat with multiple AAV serotypes, if you were to say cure somebody, could they be reinfected, and then if you had to retreat to cure again, what would that look like?
Yeah, so we've certainly seen this, we as a medical enterprise have seen this in hepatitis C cure, where a fair number of people who've been cured of hepatitis C have become reinfected and needed to go through therapy. In this case, that's a pharmacologic therapy, so there are not necessarily blockades to retreatment. The therapy as we have it now, an AAV-expressed transgene in a neuron is going to be expressed for a very long time because neurons don't turn over. The AAV is maintained as an episome. It's going to continue to make this. So we do have data that those cells are refractory to reinfection if they have that there. Now, we'd like to move in a direction where the enzyme is only there as long as it needs to be to get rid of the HSV, and then there's no reason to leave even if it's very safe. We don't want to leave it for 30 years. So in that case, there could be reinfection. Yeah, and then it's a problem if we actually did generate an immune response to the best AAV types. How do you come back with that? There are ways to get around pre-existing immunity. We've done some work, with like rapamycin that can actually work to help AAV delivery. Some people have done plasmapheresis. There are ways to get around it. If we get to that point, we will have made a lot of progress, and that'll be a problem that we'll deal with. All right, thank you very much.